Yeah, so we're going to step away from rocks for a little bit, but I'm going to be talking about the synthesis of a potential membrane component and then the self-assembly of that into a vesicle or liposome. So just to place us on the timeline that we've been talking about, formation of the Earth 4.5 billion years ago, the present. If you like the RNA world, there's some other dates in there. But if you don't, we still need prebiotic chemistry in order to make complex molecules that could potentially go on to form life. And so here we're talking about a place where we're existing in the prebiotic soup. We have simple molecules like fatty acids, nucleobases, that kind of thing. But the question is how we go from those to sort of the more complex biomolecules like peptide bonds, and in particular, what I'm going to be talking about are the formation of double-tailed membrane components. So for instance, a phospholipid like we see today, or other membrane components like that. Now, the reactions that we have to do in order to make these compounds are often in the absence of enzymatic help, both thermodynamically and kinetically unfavorable and hard to do. And so the question is how, using prebiotic conditions, can we overcome these reactions in order to make this happen? And so in general, you need to have conditions that are favorable for both synthesis and the self-assembly. So synthesis to make the molecules, but then the self-assembly either to fold proteins, that kind of thing, or self-assemble from a lipid into a protocell or membrane of some variety. So there are a lot of different sources people talk about. A lot of them are hydrothermal vents as an energy source, or clay and mineral surfaces, which can be catalytic and orienting. What we in the Vita group choose to look at instead is another possibility, using the sun, which is a high-energy, low-entropy source, and then also air-water interfaces, which would have been widely available on oceans and lakes and atmospheric aerosols, but are also relatively gentle conditions compared to, say, like a hydrothermal vent or something like that. So with that in mind, just to give brief background on membrane components, membranes are made out of surfactants or lipids, which have a polar head group and a nonpolar tail. The polar head group is hydrophilic. The nonpolar tail is hydrophobic. That means if these molecules are dissolved in solution, they partition preferentially to the air-water interface. So even if they're relatively soluble, you're still going to get a preferential partitioning to the surface based on the hydrophobic effect. And we can monitor this surface activity and the surface thermodynamics of such molecules using one technique, which is a Langmuir trough. Basically, you deposit a monolayer on this top and potentially think about, and it partitions to the surface from an aqueous solution or something, but you can basically move these Teflon barriers and look at the surface tension and get some of the surface thermodynamics out of that. So that's one of the tools we use to look at this. And then when we're talking about sort of prebiotic enclosure types and that kind of thing, one of the more common prebiotic protocells that people make are using fatty acids, which are very prebiotically relevant molecules. They would have existed, but they have relatively complex behavior. You require a pretty high concentration of the molecules in order to get any 3D structures like micelles or vesicles. And even if you are above that critical aggregation concentration, there's quite a bit of pH dependence on what you're going to form. You form micelles when you have fully deprotonated head groups, and so that's at relatively high pHs. And then as you lower the pH until the point where you're about half protonated and half deprotonated head groups, you form vesicles. But the vesicles and the micelles are always in equilibrium with the monomer in solution, which means you end up creating fairly leaky vesicles. So if we're talking about enclosures and encapsulating reactions and things like that, you tend to lose a lot of material out of the center. And even if you don't, they tend to be relatively unstable in terms of their susceptibility to divalent cations like magnesium, which we've been talking about as them being quite important. Now, the other regime is sort of phospholipids, which are modern biological membrane components, and they're a lot more stable. They form vesicles at lower concentrations, and they're not in equilibrium with the monomer in solution, and so therefore they're a lot less leaky. But in general, there's not a good abiotic synthesis either of a phospholipid or even a double-tailed surfactant. So the big difference here is the difference between a single tail and a double tail. And so the question is there a robust abiotic synthesis of a double-tailed membrane? And so how we choose to try to build this complexity towards a double-tailed membrane is by using photochemistry. And so if we look at the early Earth solar spectrum, you notice there's a lot more high-energy material, or high-energy photons available. That's due in part because the young sun, while it was less luminous, had more UV intensity, but it's also because on the surface of the Earth there was less oxygen, or no oxygen, and no ozone shield cutting off the high UV energy. And so people usually think about this in terms of its destructive capabilities, DNA mutations and that kind of thing. But it doesn't necessarily have to only be destructive. There are molecules, and one of the molecules the Vital Lab has studied sort of most is pyruvic acid, which is actually an atmospherically relevant molecule today, whose electronic spectrum overlaps the solar spectrum. And so it can be energetically excited and go on to do photochemistry. And this is, in fact, the aqueous pyruvic acid photochemistry mechanism, a partial part of it, that we've studied. But all we need to focus on right now is this main section. So you take pyruvic acid and you excite it. We use a xenon arc lamp, so a solar spectrum simulator. And it gets excited to the triplet N-pi star state. It's not super important. But it reacts with another pyruvic acid molecule. It forms these radicals. And then the radicals can recombine to form this dimethyl tartaric acid. And so what you need to see here is that we've gone from a 3-carbon molecule to a 6-carbon molecule doing photochemistry. Then the question is, all right, how is this relevant to lipids and membranes? Well, if we take a molecule like pyruvic acid and then a different molecule like 2-oxooctanoic acid, which is basically pyruvic acid but with a hydrophobic tail, it has all the same functionality, and so we could expect that this will do the same photochemistry. And this 2-oxooctanoic acid is at least relatively prebiotically relevant. Oxoacids have been found in meteorites, and it's an 8-carbon molecule, which is also relevant. And if that's the case, and it does do the same photochemistry, then all you need to make a double-tailed lipid are water, 2-oxooctanoic acid, and the sun in order to get to this, which is a double-tailed lipid. Not a phospholipid, but a lipid. And in fact, we do see that this happens. We've confirmed it with mass spec and NMR and a few things like that. It follows exactly the same photochemistry as pyruvic acid, and we make this dihexyl tartaric acid. So we've synthesized a double-tailed lipid, exciting. But what's also exciting is we do this chemistry, we just make a solution of the 2-oxooctanoic acid in a beaker, shine light on it, and then, as we see, we go from the clear solution while the photochemistry, which is before the photochemistry happens, and then as the photochemistry occurs, we see this cloudiness. We see the solution turns opalescent. And we're, in fact, just in the course of photolysis, self-assembling into ordered assemblies. And we've started characterizing what these 3D structures are. We've done both confocal and phase contrast microscopy, fluorescence microscopy, and we see that they have a spherical shape, which indicates that they're an ordered assembly rather than disordered aggregation. We also have done dynamic light scattering to look at the size of them, and we see that they're a single type of structure. They're relatively monodispersed in size. And their radius, which is on the order of 100 nanometers, is much more in keeping with a vesicle rather than a micelle or something like that. So taking this together, we're pretty confident that we have vesicles, and they're not in equilibrium with micelles as they would be with, say, a fatty acid. And we're currently confirming this characterization with both dye encapsulation and cryo-EM experiments. But if you do think that we have vesicles, we need to talk about their stability. And so their stability to divalent cations like magnesium. These are all preliminary results, but we've seen to some extent that they do appear to be at least a little bit more robust than just fatty acid vesicles in the presence of magnesium. And then in terms of their temporal stability, so again, this is the pre-photolysis and this is the post-photolysis. This particular picture is from July 31st, 2013, which means tomorrow is their first birthday, and they're sitting in the fridge, living happily. Or not living, but sitting around. So they seem to be quite stable temporally. They don't seem to make big aggregates and separate out, that kind of thing. So what we don't really know exactly is the importance that the kinetics of this process may have. It looks like the longer photolysis time that we have for our particular thing appears to actually destabilize the vesicles. They last less long if we did a longer photochemical experiment at the beginning. And that may be in part to this minor product that we make. And I won't go into the mechanism too much, but it's you take the 2-oxo-octanoic acid, which is a starting material, and you end up making pyruvic acid, and then the two of them come together to make this methylhexyl tartaric acid. So it's a single-tailed molecule. And it comes into play at longer time scales. And so basically, we want to know what the ratio of the single to double tail might be important. And if that's the case, then maybe rather than just having double-tailed lipids, we have a mixture of double-tailed and single-tailed lipids. So then what we're most interested in terms of this and being at least reasonably physical chemists, is the role that the surface is playing. So I mentioned that surfactants partition preferentially to the surface. Well, double-tailed surfactants partition much more preferentially to the surface. And so if we're doing this photochemistry in an undisturbed reaction and they're folding up into these structures, it makes sense that the surface would have some sort of role. And if we can begin to characterize that mechanism for self-assembly, then we begin to understand, I guess, how this is happening. And the other thing to keep in mind is the role of pH. We're dealing with oxoacids, so we're in relatively acidic environments, and we're starting to look more at the changing the pH of the photochemistry. But if the protonation state is important, you might think that the photochemistry would happen more readily in acidic environments, but the self-assembly might happen more readily in basic environments. And if that's the case, we need to look at the various different water surfaces that would have been available either on the early Earth. And so you have the ocean pH, but you also have all of these atmospheric aqueous aerosols, which have a lower pH in general and undergo a lot of reactions and a lot of sort of processing in the process. So you kind of need to think about this cycle between the atmospheric aerosols and the water surface on the ocean. And so just want to thank everybody, especially Elizabeth Griffith, who started off on this project. So.
Questions?
So I was wondering, a lot of the problems with prebiotic chemistry is a matter of yields and kind of dilution in an infinitely large ocean. So I was wondering what concentrations you're working at, if they're near the critical micelle concentration, and if that's prebiotically plausible.
So we work with about 6 millimolar 2-oxo-octanoic acid, which is at about the solubility limit of that molecule. It's nowhere near, we don't ever see any micelles in the precursor, so we're not close to the critical micelle concentration. And for similar acid of the same length, it's 145 millimolar or something like that. So it does seem to be that the double-tailed is exceeding the critical vesicle concentration more than the single-tail. In terms of the concentration overall, that's where maybe the atmospheric aerosols come into play, because if you're partitioning the surface, you're going to get an enhancement in concentration. And also, if you're in an aerosol, you've got a much higher surface area to volume ratio in general, and you can evaporate some of the water off.
Great, so let's all thank Rebecca again for that talk.